Hello students, today we are going to study another topic from first year biology, nucleus. Nucleus is singular and nuclei are plural. This is the diagram of nucleus. In the case of animal cell, nucleus is present at the center of the cell. On the other hand, in the case of plant cells, it lies at one side. But remember one thing, that nucleus, any substance that is present at the center of the cell, we call it the nucleus. So it is present at the center, we call it the nucleus. But when we study plant cells, there is a little bit differences in between the position of nucleus in plant cells and animal cells. In animal cells, it is present at the center, but in the case of plant cells, it is present at one side due to the presence of a large structure that is present at the center, we call it the central vacuole. Now, central vacuole occupies the center region of the cell. So, in the case of plants, nucleus at present at one side. Nucleus was discovered by Robert Brown in 1833. It regulates overall activities of an individual cell. J. Hammerling in 1934 demonstrated on the basis of his nuclear grafting experiment with the algae. Acetabularia cerenulata and Acetabularia mediterranea. The nucleus determines the morphological character of the cell and ultimately of the individual. Although universally present in all kind of cells, it means nucleus is present in all cells. Well organized nucleus is generally absent in the case of bacteria, and you know that bacteria are prokaryotes organisms. And in the case of prokaryotes organisms, there is no organized nuclei are present. And in the case of blue-green algae, organisms in which nuclear material is not bounded by a definite nuclear membrane are classified as prokaryotes. Here the nuclear material is called incipient nucleus. Incipient nucleus, it means organophore. Organisms with definite nuclei bounded by nuclear membranes are called eukaryotes. Eukaryotes, now you know that organisms having definite membrane bounded nucleus is present called eukaryotes. The nucleus may even be absent in some cells, just like RBCs and sieve elements of phloem. Mature RBCs, red blood corpuscles, and the scientific name of red blood corpuscles erythrocytes, and sieve elements are sieve tubes of phloem. Phloem is the food conducting tissue in the case of plants, and phloem is a complex tissue. It means in some eukaryotic cells, nucleus is absent, mature RBCs and sieve tubes of phloem. Shape. When we study shape, the shape of the nucleus is variable. The most usual shape of nucleus is spherical. It is a spherical in shape or oval in structure, ellipsoid or discoid in ciliates. The nucleus shows a variety of shapes. It is sheath-shaped in vorticilla, kidney-shaped in paramecium, and bead-like structure in the case of spirostomum. These are the shapes in the case of this are A, B, C, D1, D2, D3 and D4. These all are shapes of nucleus. When we study shapes of nucleus, different shapes of nucleus in animal cells. A is a elongated in muscle cells. This is a A structure, this is a elongated. Nucleus is, the shape of the nucleus is long in the case of muscle cells, and this B structure, it is a lobed in the case of human neutrophil cells. In B structure, you can see here in the B structure it is a lobed, in the C structure branched in a silk spinning cells and insect larva D1 to D4 variable shells shapes in the case of leukocytes. Leukocytes, as you know that leukocytes is a scientific name for WBC's red blood corpuscles. Now the size of nucleus. The average diameter of nucleus varies between 5 to 25 micrometer. However, the size varies with the size of the cell. The size of the nucleus depends upon the volume of the cell, amount of DNA and protein and metabolic state of the cell. The size depends upon the volume of cell, amount of DNA, proteins and metabolic state of the cells. According to Harvey, the size of the nucleus of a specific and can be derived by using following formula. You can note this is a formula of the size. When we study number, generally cells are uninucleated. Uninucleated means having one nucleus. Uni means one and nucleated means nucleus, having one nucleus. Some cells have two nuclei are called binucleated, paramecium. In the case of paramecium, we have found two nuclei. One is small nucleus and other is large nucleus. Small nucleus and large nucleus in the case of paramecium. Hepatocytes, chondrocytes. Hepatocytes, these are the cells of hepatic cells, or we can say these are the liver cells. And in the case of chondrocytes, in certain cells nuclei may be present, they are called multinucleated, many nuclei are present, called multinucleated cells. For example, in ciliate, in algae and in the case of bone marrow cells. In animal, multinucleated cells are syncytial cells. These are found due to the loss of cell boundaries during the course of development. The multinucleated cells of plants represents xenocytic. In the case of fungi, we use the term xenocytic. This is the structure of nucleus. When we study the structure of nucleus, the nucleus is composed of these structures: nuclear envelope, it is also called nuclear membrane, nucleoplasm, chromatin fibers, also called chromatin network, and nucleolus, are also called plasmosome. First, let us deal with nuclear envelope or nuclear membrane. Nucleus is externally surrounded by a nuclear envelope or nuclear membrane or karyotheca. It is composed of an outer and inner membrane, each of about 75 to 90 angstrom in thickness. The two membranes are separated by a perinuclear space or cristae or cisternae of 100 to 150 angstrom width. Sometimes the outer nuclear membrane bears ribosomes on it. It also shows structural continuity with the membranes of endoplasmic reticulum, Golgi bodies and lysosomes. And you know that the outer membrane having ribosomes, and ribosomes are attached with the rough endoplasmic reticulum. Is a continuity with the help with the endoplasmic reticulum, Golgi complex and the lysosomes, and these organelles, endoplasmic reticulum, Golgi complex and the lysosomes, collectively called germ system of the cell. The inner membrane, when we are talking about, this is a structure of nucleus. You can see in this diagram, these are the ribosomes over the presence of outer membrane, outer nuclear membrane or outer nuclear envelope. You can observe these ribosomes, dot-like structures are actually, these black dots are actually ribosomes which are present over their surface. When we study the inner membrane, inner nuclear membrane is internally supported by a fibrous structure, uniform thickness 300 angstrom, which is known as fibrous lamina or internal dense lamina. The nuclear envelope is perforated by many circular or octagonal pores, and these are called nuclear pores. When we study nuclear pores, the number of nuclear pores varies according to the species and even type of the cell in the same species. Usually their number varies between 40 to 150 per square micron, with center to center pore distance being about 1500 angstrom. The nuclear pores are plugged by hollow cylindrical structure called as annuli. The pore and annulus together form a pore complex, which is an octagonal in structure. The annuli have an outer diameter 1200 angstrom and inner diameter 400 angstrom. The nuclear pores are pathway for the exchange of macromolecules between cytoplasm and nucleus, which is facilitated by a specific protein, nucleoplasmin. Now, this is a function of nuclear pores, that it helps in a direct communication or exchange of macromolecules. Now, exchange of macromolecules, what it means? It means transfer of RNA from nucleus to cytoplasm during the protein synthesis. RNA that is formed inside the nucleus, then after the formation it transferred towards the cytoplasm through the nuclear pores. And here this is a point, this macromolecules between cytoplasm and nucleus, which is facilitated by a specific protein, is a name of protein, the nucleoplasmin. Nucleoplasm, it is also called karyolymph. The nucleus is filled with transparent, semi-solid, jelly-like granular acidophilic ground substance matrix are known as nucleoplasm. It is also called karyolymph or nuclear sac. Chromatin fibers and nucleolus are suspended in it. It has a very complex composition. Its main constituents are nucleic acids. Nucleic acid, you know very well about nucleic acid. In the case of nucleoplasm, nucleic acids are DNA and RNA. Nucleoprotein, the protein actually in the case of nucleoplasm there is a basic protein, nucleoprotamines. These are the proteins which are present in the case of nucleoplasm. These are the proteins, enzymes, minerals are present in the case of nucleoplasm. Nucleohistones. Histone is a actually name of protein that is always associated with the eukaryotic DNA, and nucleohistone protein is absent in the case of prokaryotic DNA. These have high molecular weight, 10,000 to 18,000 Daltons, and are rich in basic amino acid arginine, lysine and histidine, but lack tryptophan. The histone proteins remain associated with the DNA, prevent its transpiration into RNA. In eukaryotes, 5 types of histones are identified as H1, H2A, H2B, H3 and H4. In RBCs, separate histone H3 is also reported. Next is the acidic proteins. These are usually high molecular weight and rich in acidic amino acids, tryptophan and tyrosine. These are also called non-histone proteins. They may be linked to DNA or remain free in nucleoplasm. The most abundant DNA linked non-histonic protein is phosphoproteins. Phosphoproteins are described by these two scientists, Kodenau and Levine in 1974. Non-histone proteins are continuously synthesized during all the phases of cell cycle, while histone proteins are synthesized only during S phase. Now, S phase, it is a subphase of cell cycle. In the case of interphase, during interphase, this protein histone protein is synthesized. These proteins are synthesized in the cytoplasm and thereafter transported into the nucleus. Enzymes. The nucleoplasm contain many enzymes which are essential for the synthesis of DNA and RNA. These enzymes are made up of non-histone acidic proteins. Some of enzymes found in nucleoplasm are DNA polymerase, RNA polymerase, ATPase, adenosine deaminase, aldolase, pyruvate kinase, etc. These are the name of enzymes. DNA polymerase, RNA polymerase. These are the common enzymes used in the case of transcription, translation, or DNA replication or transcription in the case of nucleus. It also contains several cofactors and coenzymes. We have found minerals also in the case of nucleoplasm, several inorganic ions, for example, phosphate, potassium, sodium, calcium, and magnesium also present in nucleoplasm. Chromatin fibers. The nucleoplasm contains several fine thread-like basophilic structures scattered in it. This is also called chromatin network, and these are the network of threads we call it as a chromatin network. This can be seen only during interphase. Interphase, as I already told you, that interphase is the phase of cell cycle. During mitosis or meiosis, these threads become thick and conspicuous, and then these threads are called chromosomes. On the basis of staining properties, two types of chromatin material have been identified as heterochromatin and euchromatin. Heterochromatin, these are dark staining and contains region of chromatin which can be identified by acetocarmine or fullerene stain. These regions are sometimes called chromocenters or karyosomes or false nucleoli. Heterochromatin occur around the nucleolus and at the periphery of nucleus. It contains relatively small amount of DNA and large amount of RNA. Therefore, it is supported by metabolically and genetically inactive. Heterochromatin is two types, constitutive and facultative. Constitutive heterochromatin is found in those regions of chromosomes which is proximal to centromere and are constant, thus serve as a chromatin makers. Constitutive DNA is highly repetitive and it was originally called satellite DNA. Facultative heterochromatin is represented by sex chromosomes which becomes heterochromatic only at certain ages. Euchromatin, it is the light staining and diffuse region of the chromatin. It contains relatively large amount of DNA, is genetically as well as metabolically active part of chromatin. Means we have found two types of chromatin, heterochromatin and euchromatin. Inshallah, in next class we will study about the nucleolus. Thank you for the watching. Please subscribe and share.